Hi everyone, welcome to Unit 4 on genetics. So here is our overview of Unit 4. You will see that we are starting off today with mitosis and meiosis, and we will get into some Punnett squares, inheritance. And for Unit 4, just to keep in mind, there's a lot more application. So as we go through the notes, make sure to take good notes, okay, especially on those practice problems because you will be doing those in class. All right, so we're going to go ahead and get right into 4.1. Make sure to have your notes and fill in your new vocabulary. You do have a new vocabulary list for Unit 4. Okay, so the first part of this is all review from mitosis before the break. So this stuff right here, I'm just going to go over very quickly because by this time we've seen this at least 2 or 3 times, okay? So remember, chromosomes were the thread-like structures of DNA and protein that contain genetic information. Okay, here with some of that terminology, and once again, we'll just go ahead and look at this picture together. So this whole thing here is the chromosome, okay? The chromosome is composed or made of condensed chromatin, okay? So here, all that condensed chromatin is what makes up the chromosome. The chromatids, okay, here are kind of like the legs. So this is one chromatid here, and this is one chromatid, okay, so the legs. The centromere is what joins the chromatids together. The telomeres were the ends, okay? And that was our quick review on chromosomes. So the cell cycle, okay, we talked about the eukaryotic cell cycle. Our main focus today is just going to be reviewing mitosis. If you recall, the cell cycle had our interphase, phases of growth and DNA replication. S phase, remember, was that DNA replication. And then we have our M phase. And within the M phase, we had mitosis and cytokinesis. All right, so quick review on interphase. Okay, now the M phase, or mitosis. So this was that last lesson that we covered right before break. Okay, so this is the part of the eukaryotic cell division during which the cell nucleus divides. And here we have PMAT, our 4 stages. So we have prophase, metaphase, anaphase, and telophase. Now, why are we reviewing this? Because mitosis and meiosis are very similar in their stages. So if you're able to recognize the stages of mitosis, you should be able to recognize the stages of meiosis. So this first one, prophase, first and longest phase, genetic material inside the nucleus condenses and the chromosomes become visible. Metaphase, okay, this is the phase of mitosis in which the chromosomes line across the center. So remember, metaphase, think middle. So if you see chromosomes lined up in the middle, we know that that is metaphase. Anaphase, phase of mitosis in which the chromosomes separate and move to the opposite ends of the cell. So for anaphase, remember, you are thinking apart, right? So these chromosomes are now being split apart to prep for telophase, and that's the phase in which the distinct individual chromosomes begin to spread out into a tangle of chromatin, and this is the last phase that occurs before cytokinesis. And cytokinesis is the division of the cytoplasm. And if you recall, animals and plants, that happen just a little bit differently. Okay, so that was a really quick review, but remember, mitosis is the part of the eukaryotic cell division during which the cell nucleus divides. And mitosis, remember, the whole point of it was just to make new cells, right? We have constant cell turnover. We are always needing to produce new cells, and the whole point of it is to produce a genetically identical, okay, cell in comparison to that original parent cell. Now this diploid and haploid thing, we're going to discuss that in just a moment. Now meiosis is the process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. And meiosis produces haploid cells. Some people also say meiosis, I say meiosis, you can say it either way. But this is going to be what we look at today. Okay, so we'd gone over chromosomes, the structure. Okay, we know that those are strands of DNA and protein inside the cell nucleus, and we know that they carry genes. Now, homologous chromosomes, we haven't heard this yet, but homologous chromosomes are whenever you have one set that comes from the male parent and one set comes from the female parent. Diploid is a cell with 2 sets of homologous chromosomes, and that should be easy to remember because whenever you see diploid, di, think 2. The diploid cells of most adult organisms contain 2 complete sets of inherited chromosomes and 2 complete sets of genes. So for example, if an organism inherited 2 chromosomes from each parent, their diploid number would be 4, okay, or 2n equals 4, because Again, 2N is representing that diploid. Haploid is a cell that contains only a single set of chromosomes. If an organism inherits 2 chromosomes from each parent, their haploid number would be 2, or N equals 2. So really, if you just divide this number 4 by 2, that would get you the haploid number. So if you know the diploid number, you can find the haploid number and vice versa. So here's an example, okay, we are humans, right? So our haploid number is 23 and our diploid number is 46. We know that humans have 23 pairs of chromosomes, or 46 total chromosomes in all of their diploid cells. Humans have 23 chromosomes in haploid cells, which include our gametes, and we're going to find out that gametes are sex cells. The diploid cells of most adult organisms contain 2 complete sets of inherited chromosomes and 2 complete sets of genes. So if you're looking at this and you're kind of confused, here's kind of the main idea as we go through meiosis. Okay, mitosis again produces just genetically identical cells. We are replacing cells. Meiosis is going to produce sex cells, or gametes. Okay, so think of it like this. You start off the gametes that whenever the egg and the sperm combine, they both have 23 chromosomes, right? So egg has 23, sperm has 23. Whenever those two come together, then we get 46 total chromosomes, or 23 pairs, which is what we need to function and be human beings. So that's why we're talking about this whole diploid and haploid number. All right, so first check for understanding, okay? So go ahead and pause here and try this on your own. Okay, so for the fruit fly, Drosophila, N is equal to 4. Whenever we see N, we are automatically thinking haploid, right? Haploid N. What is the diploid number? So if N equals 4, just multiply that by 2, and we get 8 for the diploid number. For number 2, an organism where 2N, okay, 2N, we are automatically thinking that is diploid, okay, equals 18. What is the haploid number? So if 2N equals 18, we simply divide 18 by 2 to isolate and get that N, and we find out that the haploid number would be 9. Number 3, if an organism has a diploid number of 10, so we're thinking in our head, okay, diploid number, that is 2N equals 10. What is the haploid number of the organism? So if 2N equals 10, then the haploid number is N equals 5. Okay, so now we're going to get into the phases of meiosis. There's a lot of information here, but the good news is, for the most part, it looks really similar to mitosis. So again, if you understand PMAT for mitosis, you will understand this for meiosis. Now, meiosis, we have 2 phases, meiosis 1 and meiosis 2. So what happens? Before meiosis 1, chromosomes are replicated, producing sister chromatids. During prophase 1, we have each replicated chromosome pairs up with its corresponding homologous chromosome, forming what we call a tetrad, and that allows for crossing over, okay? So whenever you hear crossing over, the main thing that you need to remember is that it occurs in prophase 1. That's the one important thing. And the second thing is that it produces new genetic combinations, okay? So here, all right, we have your chromosome pairs. Here we have this, what we call tetrad, okay, as they are kind of joining together, forming, and then crossing over, again, is the corresponding segments of the homologous chromosomes cross over and exchange sections to produce new genetic combinations. So here, these 2 chromosomes had different genetic combinations than these 2 did, okay? Then we get to metaphase and anaphase 1. Okay, metaphase, we know that they line up in the middle, right? Always think meta middle. Anaphase, we know that they homologous chromosomes are then pulled apart. Okay, anaphase apart. And then telophase and cytokinesis, the separated chromosomes are enclosed in a nuclear membrane, and then the 2 cells are formed. Okay, so by the end of meiosis 1, what you need to remember is 2 daughter cells are produced, and they each contain a unique combination of chromosomes. Now for meiosis 2, before it happens, the chromosomes are not replicated. We don't need to do that because that already occurred before meiosis 1. Now, during prophase 2, we don't have to have crossing over, right? It already occurred. We already have that new genetic combination. And instead, each chromosome, okay, at this point, consists of 2 chromatids, and they are no longer genetically identical due to the crossing over that occurred in prophase 1. Now for metaphase, anaphase, telophase, and 2, and cytokinesis, you notice all of this is kind of compact together because it looks really similar to what already occurred. Okay, the only difference is what's happening between the homologous chromosomes and now the chromatids. So the final 4 phases of meiosis II are similar to those in meiosis I, but the result is 4 haploid daughter cells. So here we have metaphase, K lining up, anaphase. Now you notice that these K chromatids are pulled apart. Worse, and here it was the homologous chromosomes that were pulled apart. Okay, so anaphase 2 pulled apart sister chromatids. And then after, at the end, okay, we can kind of look at this picture here. The main point of meiosis II is that we now have 4 haploid daughter cells. Okay. Oops. All right, check for understanding. Go ahead, pause here, and try this on your own. Okay, so 3 differences between meiosis I and meiosis II. You could have written more. Here we wrote 4. Okay, and yours can be phrased a little bit differently. For number 1, remember crossing over occurs in meiosis I during prophase I, but it did not occur in meiosis II. Number 2, it was the homologous chromosomes that separated in meiosis I, whereas in meiosis II, it was the sister chromatids that separated. And remember, these started to be pulled apart during anaphase. Okay, anaphase apart. Number 3, what happens at the end of them? Meiosis I produced 2 daughter cells, while meiosis II produces 4 haploid daughter cells. And number 4, DNA is replicated before meiosis I, but it was not replicated before meiosis II. Okay, so again, what's the whole point of this? Mitosis, meiosis I and II. We have to have haploid cells to lead to the production of offspring. Okay, without meiosis, the number of chromosomes would double every generation, which would be a bad thing. We don't need that. So haploid cells allow for the production of gametes. Okay, and gametes again are sex cells, so sperm in males and eggs in females. So again, for humans, if we're thinking about this example, the sperm in males contain 23 chromosomes. The eggs and female contain 23 chromosomes. Okay, those are both haploid cells. Whenever those two join together to make you and us, then we get a total of 46 chromosomes, okay, or 23 pairs. And again, that's what, like humans, us, we need to function. Okay, a zygote is a fertilized egg formed from the combination of 2 gametes. Now we're going to compare mitosis and meiosis. So the 3 major differences between mitosis and meiosis. Again, mitosis produced 2 genetically identical daughter cells, while meiosis produces 4 genetically unique daughter cells. So here we have this. Okay, here we have the products down here. In anaphase of mitosis, the sister chromatids separated, while in meiosis, homologous chromosomes separate first. That was during anaphase I. And the sister chromatids did not separate until anaphase II. Okay, and then number 3. Mitosis does not change the chromosome number. Again, the whole goal of mitosis was just to produce genetically identical new cells, okay? While meiosis results in the cells with half of the original number. All right, so we have reached our exit ticket. Go ahead, pause here, and try these on your own. Okay, so number 1, haploid number of 6. So we're thinking in our head, n is equal to 6. Then what would 2n equal? So 6 times 2, we get 12. Number 2, crossing over occurred during prophase I of meiosis. Okay. Number 3, the illustration to the right represents what stage? So this one's pretty hard, okay, if you're just looking at it, but let's kind of look at this together. So we see that they are being pulled apart, right? They're not lined up in the middle, and they are definitely attached to those spindle fibers being pulled apart. Now the key here is it chromosomes being pulled apart, like the homologous chromosomes, or is it the chromatids? Since we can still see here the chromosomes, they are still clearly connected, we know that this is anaphase I. If it was the chromatids being pulled apart, that would have been anaphase II. Number 4, unlike mitosis, meiosis results in the formation of 4 haploid gametes. And number 5, last one, fertilized egg was called a zygote, and if you remember, the zygote was due to the combination of 2 gametes. So that's the end of lesson 4.1. Go back, double check your vocabulary, make sure that you have your list ready to go, and thank you for watching.